Today's global challenge of fossil fuels depletions and environmental concerns urges us to explore sustainable energy solutions. So hydrogen emerges as a promising renewable energy source due to its high energy content, zero emissions, and versatile applications in the industry, transportations, and electricity generations. So microalgae offer a sustainable pathway for hydrogen productions through anaerobic digestions, photofermentations, as well as dark fermentations. Microalgae grow rapidly and thrive in diverse environments without competing for lands, making them an ideal bioenergy feedstock. They use sunlight, water, and carbon dioxide to potentially produce hydrogen, aligning with our pursuit of clean energy solutions. Microalgae offer a sustainable pathway for hydrogen production through anaerobic digestions. So anaerobic digestions for hydrogen productions from microalgae involves enzymatic breakdown of organic molecules by bacteria, fermentations into hydrogens and acetic acid, and inhibitions of metagenesis to maximize hydrogen's yield. So the first stage is hydrolysis, where hydrolytic bacteria release enzymes that catalyze the breakdown of complex organic molecules such as protein, lipids, and polysaccharides into simpler forms, sugar, amino acids, and fatty acids. So the key enzymes like cellulases, hemicellulases, proteases, and lipases play crucial roles in this initial stage. They prepare the organic materials for subsequent conversion. The second stage is acidogenesis. So following hydrolysis, acidogenesis ensues. Here, acidogenesis bacteria ferment the simplest substance like sugars, amino acids, and fatty acids, producing a spectrum of compounds, including alcohols like ethanol and methanol, ammonia, carbon dioxide, and volatile fatty acids such as acetic acids and butyric acids, propionic acids, and valeric acids. So optimal conditions including a pH range of 5.5 to 6.5 and sufficient hydraulic retention times are crucial for maximizing fermentation's efficiency and biogas productions. So the third stage is acetogenesis. Acetogenesis further refines the fermentation's product. Acetogenetic bacteria such as Clostridium acetobutylicum and Acetobacterium woody convert volatile fatty acids and alcohol into acetic acids, hydrogens, and additional carbon dioxide. This stage thrives under anaerobic conditions and benefits from a high hydrogen concentrations, which inhibit metagens, which is microbes responsible for methane productions and thus promoting hydrogen accumulations. The final stage in anaerobic digestion is metagenesis. However, metagens convert hydrogens and acetic acids into methane, a process we seek to mitigate and hydrogen production scenarios. Metagenic archaea operate in two main types: acetoclastic metagens, which directly convert acetic acids into methane and carbon dioxide, and hydrogenotrophic metagens, which utilize hydrogen and carbon dioxide to produce methane. To prevent methane formations and preserve hydrogen, strategic measures are employed. It is crucial to halt metagenesis for hydrogen productions. So to halt metagenesis, inhibitors like 2-bromoethanosulfonate and nitrate are introduced. BES functions by mimicking coenzyme M, a critical component in metagenic pathways, thereby blocking the enzymes responsible for methane productions. Nitrate, on the other hand, acts as an alternative electron acceptor, competing with metagens for electrons and diverting pathway away from methane productions through denitrifications. For a successful anaerobic digestion, these critical operational methods are important to maintain. First, pH must be maintained at 6.5 to 7.5 and temperature controlled mesophilically 35 to 40 degrees Celsius or thermophysically 50 to 60 degrees Celsius. Hydraulic retention time is important to regulate substrate durations in the digester. As for nutrient balance, it adequate nitrogen, phosphorus, and trace elements for microbial growth and enzymatic processes. The advantages and limitations of this process is that Its advantages of microalgae-based anaerobic digestion include rapid cultivations and efficient biomass conversions to hydrogen. However, precise environmental control and biomass pre-treatment are essentials for optimal performance.
Hello and Assalamualaikum. My name is Muhammad Ifan Ashraf and I'm going to explain my part, which is the duct fermentation. Duct fermentation is an anaerobic biological process in which microorganism breaks down organic matter to produce hydrogen gas. Unlike other fermentation processes that require light, duct fermentation occurs in the absence of light, making it a versatile and sustainable method for biohydrogen production. The integration of microalgae biomass as the feedstock for duct fermentation has garnered significant interest due to the high carbohydrate content and rapid growth rates of microalgae. Microalgae can be cultivated using non-arable land and saline of wastewater, making them a sustainable and environmentally friendly option for biohydrogen production. Furthermore, the use of microalgae not only enhances the sustainability of duct fermentation, but also contributes to bioremediation of wastewater and the reduction of carbon dioxide emission. Hydrogen is a clean energy carrier, and its production through duct fermentation using microalgae feedstock align with the global need for renewable energy sources and the reduction of greenhouse gas emissions. Hydrogen produced in this way can be used in fuel cells to generate electricity with water as the only byproduct, providing a promising alternative to fossil fuels. Next, duct fermentation involving microalgae utilize their biomass as a feedstock for anaerobic microbioprocesses to produce hydrogen. The mechanism typically involves several stages. First of all, the hydrolysis. Enzymes secreted by microorganisms or released from microalgae break down complex carbohydrates such as starch and cellulose into simple sugar, which is glucose, fructose, and other organic compounds. Next, we have the fermentation. Fermentative bacteria further metabolize this sugar into organic acids such as acetic acid, lactic acid, alcohols, and gases, which is hydrogen and carbon dioxide. This stage is crucial for hydrogen production. Thirdly, hydrogen production. A specific microbial pathway within the fermentative process, such as the butyrate or ethanol acetate pathway, lead to the production of hydrogen as a metabolic byproduct. Duct fermentation utilizing microalgae biomass offers several advantages in the context of biohydrogen production. Firstly, the high carbohydrate content. Microalgae are rich in carbohydrates such as starch and cellulose, which serve as excellent substrate for duct fermentation, enhancing the potential for hydrogen production. Next is rapid growth rate. Microalgae have fast growth rates compared to traditional crops, allowing for quick biomass production and continuous feedstock availability for fermentation process. Thirdly, the versatile cultivation. Microalgae can be cultivated in various environments, including non-arable land, sand, water, and wastewater, minimizing competition with food production and enhancing sustainability. Lastly is the carbon sequestration. Utilizing microalgae for duct fermentation is not only produce biohydrogen but also facilitate CO2 capture from the atmosphere during their growth phase, contributing to greenhouse gas mitigation. These advantages underscore the potential of duct fermentation using microalgae as a sustainable and efficient method for biohydrogen production, aligning with the global efforts toward renewable energy and environmental sustainability. While duct fermentation utilizing microalgae biomass for biohydrogen production offers numerous advantages, there are also several notable disadvantages. Firstly is the low hydrogen yield. One significant drawback is the relatively low yield of hydrogen compared to other biohydrogen production methods. This limits its efficiency and competitiveness in commercial application. Next is the complexity in process optimization. Duct fermentation process involving microalgae require precise control of various factors such as temperature, pH, and substrate composition to optimize hydrogen production. Achieving optimal condition can be challenging and resource intensive. And lastly, the high cost of production. The cost associated with cultivating and maintaining microalgae cultures, as well as the energy-intensive process of fermentation, can be considerable, impacting the economic viability of the technology. These disadvantages highlight the challenges that must be addressed to enhance the efficiency and feasibility of duct fermentation using microalgae for biohydrogen production. So that's all for my part for the duct fermentation. Thank you.
Hi, my name is Sherin Anatiko, and now I will present about the last point, which is photofermentation processes by microalgae for hydrogen production. This process of photofermentation is occurring in the presence of light and then transform organic molecule into biohydrogen and carbon dioxide. This process utilizes solar energy rather than glucose as the source of energy. As it is a fermentation process, it involves an anaerobic processes. Why we use microalgae in photofermentation? There's a lot of reasons, such as microalgae have a much higher growth rate. Microalgae also have the capacity to resolve carbon dioxide. It also possesses the ability to generate a broad range of organic compounds, which is easily accessible and cheap to obtain. Next, I will explain briefly the mechanism of photofermentation. Firstly, hydrogen is generated by a photosynthetic bacteria through the breakdown of organic compound with the aid of nitrogenase enzyme, the nitrogen deprived condition. Then, this enzyme will transform atmospheric nitrogen into amino ions that are utilized primarily as the nitrogen source of bacteria. This enzyme can be found solely in cyanobacteria as well as non-sulfur purple and green sulfur bacteria. These bacteria can obtain the energy through acetic and ATP. Then this may result in electron transfer via ferredoxin to the enzyme nitrogenase and thus result to nitrogen dioxide fixation. In the absence of nitrogen, nitrogenase enzyme proton change to hydrogen. Next is the mode of operation of photofermentation. A significant amount of photofermentation research is centered with the batch method mode of operation. This method enables researchers to find out in general the difference between microorganisms and also the operational parameter as well as the substrate. For the carbon sources utilized, the goal has mainly been on pure carbon sources retained in sterile fermentation media in order to discover optimal conditions depending on the operating choices selected. Next is the advantages of photofermentation by microalgae. Photosynthetic bacteria such as microalgae can utilize a variety of spectral energies and substrates which are capable of treating wastewater from dark fermentation. This process also inhibits greater substrate transformation effectiveness and thus produce higher level amount of hydrogen. Other than that, it possesses high substrate transformation efficiency and it just needed water for microalgae cultivation. Lastly, there are few disadvantages of photofermentation of microalgae. This process has demand for the light sources as the photosynthesis effectiveness is directly proportional to its accessibility of light. It also requires a vast space in order to conduct the procedure. This process also requires a special anaerobic bioreactor in order to obtain optimum hydrogen production. As conclusion, nowadays, the global society is getting a lot aware of hydrogen potential as a temporary energy carrier during the preservation and transportation of sustainable energy, as well as its use as a natural energy for fuel cells that produce nearly no greenhouse gas emission. Now, biohydrogen is acknowledged as a potential biofuel due to its ability to possibly reduce greenhouse gas emission from the energy and transportation industry and also help to minimize our dependency on fossil fuel. Thus, a better research needs to be done in order to improve hydrogen production from microalgae for future uses.